Okay, in my little book for today, yeah, this is my diary. I don't hear any shit about it. It says, today, bio-gen demo in the fashion of 101, starting with the gene, which is where we ought to start. The purpose of giving this comprehensive primer on biology is not just for your information. It is to elucidate the analogous nature of genetic coding in the nervous system to the praxis neural network. So, biology. In elementary, middle school, even high school, usually you start off kind of learning anatomy, parts of the body, body systems. I really think they ought to teach genetics and evolution first. You don't have to teach the complexities of genetics, but at least the principle of evolution and the causality between genes and the form the body takes and behavior and all that kind of stuff. I think it would make for a more educated populace because they would understand the more complicated science once they got to high school and wouldn't have to get a PhD to to feel like they can understand it. So anyway, biology. Biology for the evolutionary biologist or the molecular biologist is genetics. Evolution is defined for an evolutionary biologist as changes in gene frequency. Changes in gene frequency. Um, all that means, loosely, is that through successive generations, um, you observe that there are a different number, different population size of a certain gene, or that the genes are found in different company, in different combinations together. That's a change in the gene frequency. When that occurs, that's considered evolution or a mutation. Now, a mutation is determined as a phenotypic change that is inferred as represented, should be expressed as a change in the genotype or just the genes. So when scientists are looking for a mutation or say there's been a mutation, they are just talking about something they've seen, a change in the trait, the phenotype. However, not all changes in the phenotype are mutations. There are hereditary traits that you certainly don't consider a mutation, certainly wouldn't consider a speciation, becoming another animal. Say your mother or father has brown eyes, blue eyes, and you have blue and your sister has brown. This is probably something you remember from high school, the Punnett square, the difference between a recessive and a dominant gene or dominant trait. And here um, the genes don't get changed in their order or in their number. It's a change in how they're told to express according simply to the probability of them expressing a certain way. And that's the simplest form. Um, and I'm sure everybody's done these. It's called a Punnett square. You have four squares in this case. And you put down some letters like so, and so you have blue eyes, we'll say they're dominant, and you have brown eyes, and then you have blue eyes, and then you have this basically wild card trait, and they combine in a matrix. You get blue eyes, blue brown, brown, and brown brown. So if there's a big B being for blue eyes, the child's going to have blue eyes. So you see in the case of the dominant eye color gene against the recessive eye color gene, out of 4 times, the dominant gene wins out 3. God, that's boring. Okay, so that's the difference between just a phenotypic change. And there can be phenotypic changes that are occurrent in an ontogeny or just an animal's lifetime, changes from the environment, people lose their hair color changes. All these things are phenotypic because they are the observable traits. All of the observable traits, everything that we can see about all life is part of the phenotype. The genes are just the basis. So I'm glad I made notes. So let's see. Genes are, there's number one, genes. Okay. So what is a gene? The sort of the fallback definition I go with is similar to what Dawkins describes a gene as in The Selfish Gene, and it's the smallest unit of replicable code or genetic information that contributes to a certain trait expressing, that defines a certain aspect. So although it has a physical corporeal existence, a gene does, as a certain length of DNA, the way that it's understood and the way that it's dealt with and used for prediction, it's not so helpful to understand it as this physical length of DNA. Now, clinicians, people who do experiments with gene therapy, of course, they're dealing with it in a much more tangible way, but still, you're not just cutting out this little section of DNA. It's much more complicated than that. What's important for us, since we're talking about information, is that not only is a gene here in DNA, let's see, DNA, this is a terrible drawing. So here in DNA, not only is it a certain length of DNA with bonds, which I'll describe shortly in it, which code for a certain trait. What it is it's the information within that length of DNA that has the potential to be expressed and is going to be acted on by environmental factors, other physicochemical elements, the length of time it has an ability to express, the length of time the organism is alive, lots and lots and lots of different factors. Okay, so that's a gene. That's genetics. A gene is just the most reducible unit that you can get to contribute to a phenotypic trait. DNA next.